Imagine having the ability to create any sort of creature you'd like. You could bring dinosaurs back to life, or maybe even a clone of yourself. Let's see what unsettling creations scientists have been cooking up in their labs. Gemini Genetics, a company in the UK, has started up a business of allowing people to clone their pets. As of January of this year, people have the opportunity to clone their fur babies when they feel that they aren't just ready to part with them yet. This has been a huge rise in demand, with many other DNA cloning locations taking in over 1,000 samples yearly. The average cost of doing these types of procedures, though, is not cheap, with the initial cost starting at £200 and the final cloning process hovering around £40,000. If you're choosing to clone a horse, though, that stacks up an extra £15,000. Is this what it's like to have money? It is a very quick process, though, and if you plan on doing this, you need to plan ahead. When your pet eventually passes away, you only have 5 days to get their DNA over to the lab. Once they have your pet's DNA, they then take a donor egg cell from a similar breed, strip it of all its own nucleus so that it only harbors the owner's pet's DNA. Then with some fancy schmancy electrical impulse machines, it boots it up to become an embryo. And then it gets replanted into a surrogate mother. Makes sense as to why it's almost 50,000 grand. I've talked about mini eyes, mini brains, and now we're getting into some mini hearts. Back in 2021, scientists were able to make organoids of 1-month-old embryonic hearts, and they also beat. Organoids are pretty much just miniature versions of our other organs, and it's much better for researchers to observe these than rather relying on animals to get their information from. These make discoveries more accurate, especially when it comes to heart attacks and why some younger folk don't scar after having one. It may also lead towards understanding underlying heart conditions. It's all created with the work of stem cells, which are cells that can be manipulated to grow into any body part that you desire. The very intriguing thing about the mini heart, though, is that it managed to grow its first heart chamber all on its own. The main goal of organoids is that you want them to be as fully functional as possible, giving you more of a chance to properly study these parts without having to get it from actual people. With the miniature heart, the fact that they get visual proof that it worked with the beating is a huge step. Even in the Bronze Age, humans were trying to make hybrids fetch. When the Kungas were first discovered alongside royals in an ancient burial site, they thought they were some extremely rare breed only given to those of higher status. But to this point, we had no idea where their identity originated from. With all the crazy modern technology that we now have, scientists were able to finally pinpoint that these strange rare beasts were actually the offspring of a female domesticated donkey and a wild male A. You know what the A stands for. Researching the remains and taking a deeper dive into their DNA has finally answered one of the biggest archaeological discoveries. They were also ancient texts discovered that they were in fact husband programs that had all the males create these Kunga hybrids. They were popular for being strong and stocky, extremely good for cultivating the land. The only problem with these texts is that they never specified what sort of animals they used to create the Kunga. Probably didn't want anyone else stealing their ideas, I guess. They were also very smart in choosing a female donkey, since they were so domesticated at the time and could easily monitor their offspring hybrids. It was a dark and stormy night, funnily enough in the month of November, right around the time the novel Frankenstein took place. Surrounding a surgeon's table in the middle of a theater, curious onlookers of scholars, doctors, and other students from the school looked eagerly at the man lying down on the table. It was clear he was no longer part of this world. Once named Matthew Clydesdale, a former inmate who was going to be the first person to showcase Andrew Ure's diabolical experiments. Holding 2 metal rods in place that were hooked up to voltaic batteries, Andrew pressed 270 volts up against the man's body, and in a shocking turn of events, the man's body shook vigorously as his arms and legs thrashed around. Andrew even added on to state, when the one rod was applied to the slight incision in the tip of the forefinger, the fist being previously clenched, that finger extended instantly, and from the convulsive agitation of the arm, he seemed to point to the direction of the spectators, some of whom thought he had come back from the beyond. Science was booming at this time, with many scientists trying to understand the connection between the natural and the scientific world, and how they would be able to manipulate that to their pleasure. Would you have the stomach to create a hybrid animal? Let me know down below, and I will see you all in the next video. Behind the scenes, scientists have been working hard at using their abilities to bring mysterious creations into existence. What do these mad creators have in store for us today? I say we put all our money down on taking out parts of the human brain, specifically portions that are believed to be linked to human intelligence, and place them into the brains of monkeys. Surely, nothing bad will happen, right? In China, scientists wanted to see if it was possible to speed up the evolution process for our primate cousins. We've all seen Planet of the Apes, right? To no one's surprise, there were a lot of noticeable changes within them, especially when they were giving neurological tests. They tested this with monkeys that didn't have the modified changes and the ones that did. And the ones that did obviously had better outcomes when they competed with tests that involved colored blocks and memory. The way their brains developed was still at a much slower rate, actually quite similar to humans in their early stages of life. So technically, we're looking at an animal that could probably learn to speak in just a few more years. Or, on the other hand, because these experiments are very fragile and extremely new, there is the possibility this could have harmful side effects later down the line. Do we really need to do this? We ain't done with monkeys yet, because now we are putting their DNA into pig embryos and bringing them to full term. Bringing anything to full term within a lab is incredibly dangerous and risky, and a lot of people claim it's highly unethical. This happened once again in China back in 2019, and they were the first group of scientists to actually manage it. Obviously, the original goal was to use human stem cells in pigs again, but there are way too many laws surrounding that out of fear that these animals are going to rise up against us. So instead, they used monkey cells and fused them into 4,000 pig embryos, and then placed these all into surrogate pigs. To focus on just one pig mother, she produced a litter of 10 piglets, with only 2 of them coming out with the monkey DNA. When they delved into some samples from these 2 little piglets, they discovered the monkey to pig ratio was 1,000 to 1 in 10,000. To put it into perspective, with the human pig experiment, that hybrid only had 1,000 in 100,000. So clearly, we're getting a lot closer in getting this ball to roll faster. Messing with nature like this though, it's no surprise that the piglets didn't last too long. This is why it's very unethical. I ain't done with brains yet either. Now we're getting into some mice with human brains inside of them. At first, all that scientists were messing around with was injecting human stem cells into them, but now we're getting a little too courageous by inserting brain organoids into their little heads. If you don't know what organoids are, they're basically miniature versions of human organs. So it's pretty close to a functional brain if it was a really early stage brain. When I say miniature, I mean lentil-sized organoids that get slipped into their tiny noggins. Right now, there was nothing out of the ordinary in terms of behavior as they continued to eat and drink, even allowing their bodies to keep sending nutrients to the organoids. It is very difficult to combine 2 very separate species into one, but with certain lab mice, their immune system doesn't reject the cells, which is why they're so popular to use. They've tried this in the past before, but for some reason those brain organoids weren't able to get much oxygen and fell apart within 5 weeks. But these new brains lasted 233 days within the mice. It got to a point where these brain organoids started creating their own neurons similar to ours, which isn't really good. At least that's just my opinion. Insect-borne illnesses are no joke, but with the help of genetic engineering, scientists are coming up with new ways to create hybrid insects to get rid of these pests. The proper term for it is called synthetic speciation, and one researcher who was working on this project stated, speciation has occurred billions of times on the planet, but hasn't been engineerable before. It's a little scary, but what they did was make an entire replica of a fly's DNA, kind of like cloning, except before it became an actual creation, they modified it and played around with its genetic coding to their desire. Hybrids don't normally reproduce, because the majority of them always come out sterile, but these flies can. They made them specifically that way. To make it even better, they modified them so that they wouldn't be able to procreate with other flies, keeping their genes very isolated and specific. Their only current worry for this is the risk of a gene drive, where these specific genetically modified genes will end up completely wiping out other genes that are not it. Would you have the stomach to create a hybrid animal? Let me know down below, and I will see you all in the next video.